Hi.
Hello, Antonio, Betty. Hello, everyone. Welcome back to our 63rd sessions of BioMed Journal Club. Today we have two speakers, and to the sake of time, I'm gonna ask Betty, my friend, to take care of the rest of the events and moderate the sessions. And Betty, please start introducing our
Thank you.
first speakers.
Yeah, great. So I'm Betty. I'm a PhD candidate at Stanford. I'm working with Sadik, and I'll be organizing the timing and logistics of this session. So our first speaker today is Dr. Emil Kristoffersen. Emil is a PhD in molecular biology from the group of Brigitte R. Knudsen in the Molecular Biology and Genetics Department of Aarhus University. He focused on nucleic acid nanostructures and their application in the detection of enzyme activity. In 2016, Emil did a postdoc in the group of Victoria Birkdale in iNano of Aarhus University in collaboration with Dr. Wilhelm Bohr at the National Institute of Health in the United States. There, he worked with the biophysical characterization of nucleic acid structures, focusing on DNA G quadruplexes and their interaction with DNA-modifying enzymes, and this was done between 2016 and 2022. This work was supported by the Lundbeck Foundation. In between these experiences, Emil, as a postdoc, worked in the group of Philippe Holliger in the Laboratory of Molecular Biology in the UK, working with catalytic RNA molecules from 2018 to 2020. And this work was supported by the Carlsberg Foundation. Now, Emil works with the design and evolution of functional nucleic acids, including RNA, DNA, and modified nucleic acids, focusing on the structure-function relationship. He uses methods such as single-molecule FRET, directed evolution, and cryo-EM to achieve this. Emil, the floor is yours.
Can you hear me? So Antonio and I are in the same room, and I will be speaking through his microphone to avoid this echo thing. I hope it's not too confusing. But I'll share my screen. Okay, so I'm going to talk to you about single molecule FRET today and G quadruplexes. And some work that we did that got published this year in Victoria Birkedal's group. So the talk is called Single Molecule FRET Reveals a Complex Folding of Consecutive Telomeric G quadruplexes. Because DNA is not something that just forms the double helix. Of course, you know, it also forms chromosomes and larger structures, but it also forms smaller structures. It forms interactions with itself, and so it forms hairpins, and it forms these quite stable, actually, G quadruplexes that forms also inside the cell. And these are something that the cell needs to regulate in order to function. Sorry. So G quadruplexes are found these years to be important in cellular health. And this talk is focusing, so there's a lot of work that's done focusing on these G quadruplexes, and this work that I've done and I present here focuses on consecutive G quadruplexes. So that is when more than one quadruplex can form next to each other. It happens both in the genomic DNA, but it especially happens in the telomeres. So just a quick overview of that. So in 2019, in 9, there was a paper in Angewandte where they used atomic force microscopy to show that consecutive G quadruplexes forms as small beads on a string. So here you have a DNA sequence that can facilitate many G quadruplexes, and they form like right next to each other. That wasn't known. But then things got a little more complicated. And now I have to move your little head so I can see what's on my screen. So here we go. In 2018, Panus and co-workers, they did a kinetic folding of this long telomeric DNA and found that, yes, consecutive G quadruplexes fold, but sometimes there's this uneven spacing between them. And this got supplemented by a work in 2021 where they used UV spectrophotometer measurements of DNA to show or to present a frustrated folding model where these DNA repeats that can lead to formation of many G quadruplexes folds in a frustrated way that I'll come back to, because my research showed the same thing but with a different method. So that's what I'm going to present to you today. So just briefly, I want to show you, because it's important then that the telomeric sequence, it's a repeat of T, TA, GGG, and it comes again and again and again, and it's made by the telomerase upon extending the telomeres. Each of these GGG repeats can form a layer in the G quadruplex, and therefore, if you have more than 4, or if you have 4, you can make 1 G quadruplex, and if you have more than that, you can make various variations of it. Okay. The work I've done and I present here is based on single molecule total internal fluorescence microscopy, where we at the single molecule level. So there we have dual labeled, so DNA molecules that has 2 dyes on, and we mobilize them on a slide, and then we excite them with lasers with this curve method, and where we create an evanescent field of fluorescence that excites the flow force really close to the surface and leaves a very low background, so we can look at them at the single molecule level, so single individual flow force on the DNA. And then by exciting the donor, if it comes close enough, I'm sure you're aware, to the acceptor, then you start seeing energy transfer and you see FRET. So I'm sure everybody has heard about FRET before, but with FRET we can then, as shown here, we can look at the folding state of the DNA. Is it unfolded? You'd have a low FRET. If it starts folding, it'll come up the FRET, and then at full folding, it'll have a higher FRET. That's at least the theory. So then we can look at single molecule dynamics at real time in real buffers, but with fluorophore labeled DNA. So another thing you should know is that this data is read out as a time trajectory. So each of these little spots, if we measure the intensity of that and calculates the FRET over time, you see these time traces, and these can be converted into histograms by just stacking all the measurements into one. And normally, so this is a small histogram made of only this time trace, but normally you put in multiple over 100 of time traces that represent single molecules measured over a certain time to get this kind of data. Okay, so let me show you what we found. So, first of all, we took a DNA sequence that could make up to 2 G quadruplexes, so it had 8 of these telomeric repeats, and it had a fluorophore, 2 fluorophores spaced in each end of it, of the DNA strand, and then it had a biotin, so we could immobilize it and use this TURF method to look at it. And as I showed you, upon folding, you'd get more and more energy transfer, and you'd see more and more FRET. Doing that with a lithium salt that we know prevents G quadruplex formation, so the quadruplexes does not form here. We saw a low FRET, so even at 30 millimolar lithium, it kind of was still straight and gave a low FRET. Upon addition of potassium or sodium chloride, both facilitates G quadruplex folding, but in 2 different ways. So potassium makes hybrids and other types of G quadruplexes. Just it can be seen, the profile of that can be seen here in the CD spectrum. If you are familiar with G quadruplexes, you'd see that this is a hybrid kind of profile. And we can also see that a lot of higher FRET populations occur now in our DNA. When in potassium, it makes anti-parallel, primarily G quadruplexes, and we also suddenly see a shift from the non-folded peak or the low Fred peak to a higher Fred peak. So definitely something is folding. So what surprised us actually was that so much of this low Fred peak persisted, and we wanted to investigate that further. So we knew already, based on a previous paper that had come out from the Victoria group by Mikael Sandrian, who was a really talented postdoc and a mentor for me when I started in the group. He did a study on DNA that had room for only one quadruplex, or not two, but only one, but still had these dyes in these positions. And he could see that if he had no salt, he had an unfolded population of low Fred, and then if he just added a little salt, 25 millimolar, it shifted up and almost folded completely. And in this paper, he shows that it forms these different conformations, but primarily the hybrid 2, which is also known from other places in the literature. That's the major folding in potassium. Now, when I did a more extensive thing, but the same thing as he did, with my double G quadruplex forming DNA substrate, I saw a much more complex folding pattern. So it's not just shifting between unfolded and primarily folded as his. I see actually a persistent, and I think I have an arrow, yeah. I see a persistent low Fred peak at both potassium and in sodium. And this was puzzling. Why does this persist? Because the quadruplex should form quickly. So why, if one quadruplex forms so easily, why doesn't two? That was the question. And I want to point to your attention on this slightly climb of the Fred in lithium. So this climb, this increasing median of the Fred observed, indicates that as you increase the salt concentration, even though the quadruplexes doesn't form, the backbone, negative charges on the backbone of the DNA is shielded by the ion, the positive lithium ion, and then the polymer kind of subtracts because it gets more affinity towards itself. So that is something that you need to be aware of when you do these salt titrations. But this is so you could look at it as a control and see where the unfolded peak would be, and then still the folded peak is much higher. So maybe it's not that surprising actually when you think about it. So we started thinking about this, and if you have a strand that can do two quadruplexes, it can also do one, but in various positions, because these repeats continue throughout. So there's 8 of the same repeats. So if you have a quadruplex nucleating in the middle, for instance, it will actually block the formation of the most thermodynamically stable form that has two quadruplexes. So we speculated that there might be a number of off-path foldings in this scheme, where only if the quadruplex forms in the extremes, they would actually lead to formation of the fully folded, but if they formed in the middle, they would actually block the formation of the fully folded. Maybe that could explain our observation of the low Fred peak we found. Yeah, so indeed, two quadruplexes is much more complicated than one. And then, as we also write in the paper, there's this little caveat that these off-path structures, it might not be as simple. There might actually be even more structures that forms and folds in the DNA, but they would actually come into this off-path category, where maybe triplexes are formed and something like that. But it still would prevent the full formation. Okay, so we designed all of these, or we designed a number of DNA constructs that were mutated in a way so it was locked in any of these, in one of these 3 positions here. So for instance, this one only had the GGGs that was needed in the positions for making number 2 here. Whereas whereas these here were different. So we could control which quadruplexes would form. And all of the ones that formed one quadruplex was actually in the low FRET range when we looked at sodium chloride. And this was actually a major find. Oh yeah, this was a major finding, and I'll show you why. I'll tell you why in a minute. But also, I want to show you that we also ran gels to validate that even though the FRET stayed low, the DNA actually did contract upon addition of sodium chloride that would make the quadruplexes form. So here we ran gels, and we can see that when you have the same amount of salt but sodium, the bands run. So the band that can make two quadruplexes run much faster when two quadruplexes formed, but also the ones that have only one quadruplex actually runs faster than the ones that doesn't. So we think that these constructs actually form and fold, but they lead to low FRET. So now we are pushing towards a model where we think that all of these conformations down here, they present some kind of low FRET regime, whereas the high FRET regime is only observed when the folding is complete and the dyes are really quite closely associated in space. So now, instead of looking at the histograms, we can actually go a level deeper and look into the individual trajectories, because we can actually look at the individual single molecules measured over time. And it turned out that in sodium, they distribute into two classes. Some of them had only low and intermediate FRET values, and some of them had high, and then sometimes jumping into intermediate values, but never too low, or very, very rarely too low. So that actually revealed for us a mechanism where we think we understand how the folding happens in sodium. So in the low FRET group, you have a population of particles that jumps between the off path and the unfolded, but they never really reach the fully unfolded state until allowing them to actually jump to the on path and then to the fully folded. And the same happens with the fully folded. Sometimes unfolds, but rarely unfolds again to get trapped. So that's what happens up here and down there. And we can then identify these states, and we can measure the length of these states. And by that, we can actually calculate the rate constants of this mechanism. But before we did that, I also have to show you a third class of particles that are really important and that actually kind of connected it all. And that is because we found between around 5% of our time traces each time that had this group escape. So they started in one group, and then suddenly something happened, and they entered the other group, and then they had they looked as the other group for the rest of the measurement time. And this is important because if we just had the two groups, it could represent two populations that never interact, and it's not in the same folding scheme. But this jump here actually revealed for us that we are looking at the same type of molecules. There's just a big penalty of actually jumping class somehow. So based on the rate constants that we got from the dwell times that I pointed out just before, we could now plot these lines, which represents a model of how these rate constants would act. They represent what would you say? They represent a model on how we think they would fit, and this fits with the data that are these, or it fits reasonably well with the data that are these black circles here. So by actually looking at the dwell times observed at the single molecule time traces, we were able to reconstruct the model, this model that we present, and here are the rate constants that we took out of that. But this was only for sodium, and the problem is actually in the cell, potassium is the more relevant ion because it's higher in the cell, and the quadruplexes that are forming in the cell is believed to be more like when you add potassium. The problem is that the situation in potassium looked much more complicated in our data. So here's just a little overview. So we also saw this low FRET peak for potassium, But and we saw this broader high Fred peak. And I don't know if you remember, but as I told you in the beginning, there's a lot of different G-quadruplexes structures that can even form in potassium. So there's a much more complicated system going on. And when we looked at our time traces, we saw the same thing. So we saw a lot of group jumping. So this conserved group, either one or the other, and then very few jumps between them, was not seen with potassium. They seem to jump group a lot of times. So likely there was another dynamics at play, but actually we thought that the mechanism was the same. But how could we investigate that? Because at the single molecule level, this is too complicated for us to solve, because a state might actually be represented by two different conformations, but with the same, leading to the same Fred. So only in sodium we were confident that these classes that we saw represented more or less the same thing. Here we weren't sure. Yeah, so this I put in to remind you that there are different conformations of G-quadruplexes forming in sodium. So what we did in the paper, and I will be happy to discuss if there are better ways. We couldn't find any, but this is a way of doing it. So we used the circular dichroism to measure this. So with circular dichroism, you can actually measure the profile of the folding of the G-quadruplex. And we could show that by adding more and more salt, you actually got a stronger and stronger profile, but just marginally, of the G-quadruplex, meaning that with low salt, there was actually a fraction of the G-quadruplexes that did not form because they were in the unfolded population. But at higher salt, more and more quadruplexes formed. And this actually is illustrated. Let me just quickly go back. In our single molecule data here, so with higher and higher salt, you see more and more folding. So this is what we expect, and this we also did see in the CD with potassium. The problem is with the CD, you are not measuring. Instead of with the single molecule, you measure those that are in the fully folded state, so you only count those that has two quadruplexes. Here you count every construct that has a quadruplex. So therefore, the method is much more noisy, because some of them will have two, but some of them will have one, and you are basically only the difference is only the difference between the quadruplexes that could have formed two but only forms one due to this strange folding pattern. However, what we did see was that in potassium, this equilibration happens instantly. So this fits with our single molecule data, that there's no energy barrier between high and low Fred. It jumps up and down already. But still there is a difference in Fred, indicating that you actually do see the same model, but not the same rate constants. With sodium, we did see a really slow equilibration of the folding, which also is supported in our single molecule data. So all in all, with that, we proposed this folding model that is actually fitting quite well with what is emerging in the literature, namely that that consecutive G-quadruplexes, they form in mysterious ways, you might say. So we think of it as soon as you let them form, they nucleate all over the place and forms these quadruplexes all over. Some of them are closely spaced, but then maybe there was a nucleation event here, and then there are some linkers in between that are not used. But what is new is we have now, so this is also what some other papers found, but what is now new is that now we have been able to actually equilibrate, so that when we look at our sample over a long time, we can see that they slowly repack and more quadruplexes form. And it moves towards this tight folding that is actually the most thermodynamically stable, we thought for a long time. But then we figured that actually, because there are so many classes of the partly folded structures, there will actually be some times where it misfolds back into the equilibrium state. So our model states that you have an equilibrium state of spaces between quadruplexes where sometimes they unfold and something refolds differently. And you would actually keep on having these spacers events emerging in a long strand that can form multiple quadruplexes. And this might actually be important for the cell, because in the cell you need proteins to load onto these regions. And with the beater on the string theory, it's been speculated that maybe the proteins cannot bind, and it's been shown that proteins cannot bind if there's a quadruplex, but maybe they actually can bind because of this more complicated model. Because if you extend the model from 2 to 3 quadruplexes, it can be even more complicated than what I've just shown you. Yeah. So in summary, I'll just say that single molecule dynamics can be measured at equilibrium, and we can produce this equilibrium by looking at non-equilibrium data, and then by combining those two, both our equilibrium data and our non-equilibrium data sets. In sodium, there is a strange trapping of structures, so it stays in either one class or the other. That is not observed in potassium, and I think that's quite interesting, something that could be investigated further. I don't know if there is something structurally in the anti-parallel G quadruplexes that makes them eager to actually unfold to an extent where they can refold and form the double quadruplex. And with that, I would like to thank the organizers, and I also would like to thank Victoria Birkedal, who is the PI of when I made this investigations. And I would also like to thank the rest of the single molecule thread group at iNano. And I'd like to thank all of iNano, and also I'd like to point you towards some other new papers that I have published and underway that you might find interesting. So thank you. And I think maybe time is good, right?
Time is good, yes.
So I don't know if you have any questions.
Emil. Yeah, so if you have any questions, feel free to unmute or leave your question in the chat, and Emil will be happy to answer.
I actually have questions since I'm sitting right next to him. I was wondering, you chose anti-parallel G quadruplex. My interest is in parallel G quadruplexes. So do you think that we can extrapolate your findings for other sequences? For
Gabriel Antonio Minero, who is sitting next to Emil, actually, and he is currently an assistant professor at the Interdisciplinary Nanoscience Center at Aarhus University. Antonio obtained his PhD degree in biochemistry from Ruhr University, Bochum, defending his dissertation entitled Electronically Controlled DNA Processing on Chip. Antonio is an expert in chemistry and biology of nucleic acids, and his research interests go beyond pure synthetic DNA. Recently, Antonio has been awarded a prestigious Willem Experiment grant for his research on rolling circle replication in phage bacterial systems. And today, Antonio will talk about the electronic pH switching of triplex DNA reactions.
Thanks for the introduction. Yeah, I have changed topic a little bit. I would like to speak about triplex DNA, including electronic pH switching of triplex DNA reactions, but under the crown of triplex forming oligonucleotides as a third strand for DNA nanotechnology. And to start my talk, I would like to remind fundamentals of DNA base pairing, and why we like DNA for nanotechnology is because of its perfect programmability. So base guanine binds base cytosine, forming 3 hydrogen bonds here, and the nucleobase adenine binds nucleobase thymine, forming 2 hydrogen bonds. This was discovered in 1953 by Rosalind Franklin and Watson and Crick. I don't remember their names, but Later, I don't know, 10, 20 years later, Hookstein has discovered another type of bonding that's now called Hookstein bonding. So now in green are the same bases that you have seen here. So guanine is still binding cytosine, and adenine is still binding thymine. But the purine bases adenine and guanine have a group that are still able to form base pairs with third nuclear base, forming triplets. So adenine can form a triplet with two thymines, and guanine can form triplet with two cytosines. However, in this case, the second cytosine must be protonated. Attention, this nitrogen in double-stranded DNA is non-protonated, but for triplex DNA, it must be protonated. So there is a possibility of triplex formation at low pH, pH below 6. And this equilibrium between double-stranded and triple-stranded DNA is pH responsive, which was a topic for my PhD degree, where I used gold microelectrodes to actuate this pH switching in electronic processing way. I would like to speak a little bit more than that, and I will start with incidence and biological relevance of triplex DNA, and we'll present triplex DNA optomagnetic biosensors. I will talk about switching of magnetic particles via triplex DNA, and culminate with using triplex DNA for self-replication controlled electronically. Triplex DNA, like G-quadruplex DNA, is common in plasmids and supercoiled genomes. This is because supercoiling introduces kinks into DNA molecules, and where there are kinks, there is a tension and pressure. So DNA unwinds locally from double-stranded DNA into two single-stranded DNA, and then it can adopt secondary structures. Among them are G-quadruplexes, zDNA, and triplex DNA. That's how triplex DNA, for example, can be found in extracellular matrix of biofilms. This is my most recent observation. So Staphylococcus epidermidis is a pathogen that forms biofilms. These biofilms are stabilized by proteins, polysaccharides, siderophores, and extracellular DNA plays a major role in establishment of this staphylococci biofilms. DNA can interact with proteins, and upon formation, there is always a flow of liquid blood vessels or movement of the tissue around the biofilm, which simulates or mimics this tension or pressure I was talking about. And I have visualized staphylococci biofilms as part of my most recent publication, in which we found G-quadruplexes that Emil was talking about, and also triplex DNA. In order to detect these structures, we used antibodies, and they were found in this web-like matrix shown in green for G-quadruplex detected with BG4 antibody, and for triplex DNA detected with Gel-466 antibody. The red spheres are staphylococci cells, and the blue is a bDNA. There was not much other DNA structures in that particular sample. What's more interesting about triplex DNA is that it can regroup into so-called H structure, or a DNA kink in a plasmid. And that was like normally it's not that logic that such a reaction would occur, but upon supercoiling, like I said, the DNA will unwind and will regroup from perfectly double-stranded into partially triple-stranded, partially single-stranded. And such kinks were detected using scanning electron microscopy 20 years ago. So this is very well known. When I moved on from about 5 years ago, I was wondering what about biosensing triplex DNA, and I found out that polypurine DNA, which is in green, is abundant in viral RNA of COVID. Well, COVID is most recently, but back then it was HIV. So viruses, they have conservative polypurine sequences in their RNA. So we can develop biosensor to detect such RNA based on triplex DNA. In this case, the sensing DNA is red, and it forms triplex DNA in response to the green target at low pH. Imagine that the magnetic particles are in a needle shape. Then in a magnetic field, they will rotate with a certain speed, creating anisotropy of light that passes through. Because in a magnetic field, the needles will be oriented in the same direction the light will pass through. If they are aggregated, they will rotate much slower, and we can catch the frequency at which we can discriminate single nanoparticles from the aggregates and detect the presence of green target, polypurine target, by aggregation of particles functionalized with red triplex-forming oligonucleotides. I won't go into the physics of the biosensor. You can find it in this paper. But we have detected this anisotropy of light passed through the suspension of magnetic particles in the presence of different concentrations of the polypurine target, obtained dose response plot, and also confirmed that the resulting signal is perfectly pH switchable. So we confirmed that this is due to triplex formation. The limit of detection of such biosensor was 100 picomolar. It might not be that low, and of course, PCR will beat us, but it's still confirming the need for triplex-forming oligonucleotides in DNA nanotechnology if we consider such fancy biosensors. What in reality we could have done, we could have amplified viral RNA using PCR and then use our triplex DNA biosensor to boost amplification to detect even lower concentration, but it was beyond our interest. The next interesting observation is that we can form sponges. Now, nothing to do with viral RNA, just we would like to build assemblies of magnetic particles for different applications, and we would like them to be switchable. And I would like to show you how we can achieve that. So we will use the same magnetic particles functionalized with polypyrimidine triplex-forming oligonucleotides, and in response to the central purine strand at low pH, they will form assembly by triplex DNA. We then will inject the excess of the green polypurine strand, and then what happens is that the green polypurine strand will bind both of the red strands, as you can see here, which will cause dissociation of the magnetic particles. So in other words, it's assemblies of magnetic particles that can be controlled, or that can sense the concentration of the central strand, and at certain concentrations they will assemble into cluster, and at high concentrations they will disassemble. This has been confirmed using microscopy. So as you can see from left to right, so here are microfluidic channels, and we inject the magnetic nanoparticles into the channels filled with a green strand. In the beginning, the magnetic particles interact with the green strand, and there are more particles than the central strand, so that they aggregate. They form aggregates, and you can see they suck out all of the fluorescence, which means they form the sponges, bright sponges aggregated via triplex DNA. They grow and grow in size by sucking fluorescently labeled green strand. But then when we inject the green strand, excess of the green strand into the microfluidic channel, you can see how they dissociate into smaller clusters, fluorescently labeled clusters, but small. So we have demonstrated on-demand formation of the magnetic clusters, dissociation of magnetic clusters, and exposed to concentration of polypurine strand in the microfluidic channel. And now I move towards the original, the title, this electronic pH switching, for which I have employed benzoquinone redox system. This benzoquinone can be recharged, so with the flow of electrons from the electrode can generate hydroxide ions in anodic mode. So when it's negative, potential is negative, it produces hydroxide ions and it increases pH. When potential is positive, it's cathode, so it uses hydrogen. I mean, it reduces pH. And from the very beginning, I was speaking about pH switching triplex, and the triplex is stabilized at low pH. So guess what? We can use these electrodes functionalized with the benzoquinone to control triplex switch. Just at 1.8 volt from a microchip, 2 by 2 centimeters approximately, we could produce these pH patterns. I mean, they are generated by the electrodes. The red electrode over here is positively charged. The blue electrode over here is negatively charged. So pH patterns, just like I said, low pH at positively charged electrode, high pH at negatively charged electrode. The pH patterns detected using SNARF fluorescence. This is a ratiometric fluorescent reporter that shifts its wavelengths, its emission from around 570 nanometers, 650 nanometers upon increasing pH. So we could detect the pH changes by ratiometric fluorescence microscopy. What we would like to do with that, after we have validated that our benzoquinone system is capable to control pH, we would like to check out if we can control actually triplex hybridization. And for that, we employed fluorescently labeled hairpin and from a oligonucleotide with a quencher. Hybridization and triplex formation, quencher binds close to the fluorescence, it goes FRET-induced quenching. Then we would reach electrodes. Some of them will be positive, some of them will be negative, and so we will be switching them between positive and negative to alternate pH between pH somewhat around 6.2 and 6.5. It's not a very big switch, I must say, but it was sufficient to alternate some population of triplex DNA. As you can see, we recorded switching of triplex by fluorescence quenching. At high pH, around 6.7, the fluorescence was highest, which means that fluorescently labeled hairpin was unbound from the quencher labeled DNA. At low pH, approximately 6.2, they were breached together and the fluorescence was quenched. When we tried to make a larger pH switch, we actually found it's challenging to reversibly switch between bound and unbound state, most likely because once pH hits really low values, something irreversible happens to this molecule. We have not investigated it further, but we found that if we switch just in between 6.2 and 6.7, off unit pH will be enough for doing something interesting with these molecules. I also found that we can switch pH remotely, because some of you may ask, oh, if you switch pH by electrodes, you also affect a flux of DNA, because DNA is negatively charged, and that's true for future applications. So we decided to see if we can recruit such hydrodynamically spatially resolved system where we could actuate electrodes in one channel and detect pH changes in a different channel. And it worked. We could detect pH changes in both actively produced and actively executed electrodes and in a channel where electrodes were not executed. There's also a reason why pH was more stable around pH 6, but it would drop rather quickly to pH around 5 or even below, is because I have employed phosphate buffer, and phosphate buffer has this pH profile, so it cannot hold very efficiently pH around 5. So once it hit pH 5.4, can easily go very low below. So what's such a wonderful assay can we do with this microfluidic chip? We implemented electrode control over triplex hybridization to induce DNA synthesis, because we wanted to build the self-replicating system. In this system, this hairpin DNA will bind the monomers or shorter sequences of triplex forming oligonucleotides. One of them has fluorophore quencher, and so fluorophore is quenched by the thread, and another will have a thiol. Well, the quencher is connected via thiol. It's disulfide chemistry that you can see on the right side, developed by Volker Patzke, my colleague from Ruhr University Bochum. So upon triplex hybridization, these two molecules bind to the hairpin. The thiol will attack the disulfide bone. The ligation will take place, the quencher will leave, and the fluorescence will light up. Then we can alternate the potential of the electrodes to switch pH between 6.2 and 6.7, so that we can sort of dissociate the product strand from the hairpin and let the excess of the building blocks to bind and to ligate again. And so what we observe in this microfluidic hydrodynamically spatially resolved structure, we observed fluorescence enhancement both in the lower channel where electrodes were executed and also in the upper channel where electrodes were not executed. So we could conclude that upon pH switching, active pH switching, we could synthesize new DNA. We could also synthesize new DNA remotely, just as a control of DNA flux. The concluding remarks of my presentation are such that the triplex DNA motifs are abundant in bacterial genomes, supercoiled plasmids, and extracellular matrix of biofilms. Biosensors based on triplex DNA can detect down to 100 picomolar of conservative polypurine RNA of viral origin. Moreover, triplex DNA assemblies are switchable both in response to pH and in response to the concentration of polypurine strands, the ones that we detect. Triplex DNA motifs were used for enzyme-free DNA templated ligation, controlled electronically in a microfluidic channel. And at last, as a perspective of my research, also what interests me currently is that triplex DNA is proposed as a scaffold for biochemical batteries because of this switching and group response of the assemblies of triplex DNA. And so they can store and release energetic molecules such as ATP, FAD, or NADH. I would like to thank all people that were involved into slides that I have shown. So this is first of all my current supervisor, Ricky Meyer, in the biofilm group. Also people from DTU, where I did my postdoc about this optomagnetic biosensor, Jeppe Volk and Mikkel Hansen. And my team from Ruhr University Bochum, which supported me in this electronically controlled page switching, is Patrick Wagler, Thomas Mack, and John McCaskill. I would like to thank all of you, and I'm ready to take the questions if you have.
I believe Vinay has a question.
Vinay, you wanted to ask something?
Oh, I was muted. Sorry. Hi, Antonio. Thank you very much for your talk. It was nice to hear back after a long time. I have actually a question related to this triplex forming probes, and my question is that if I remember that the duplex forming sequences in the triplex should have longer sequences of A and T types bases to form these stable triplexes. But in the context of, for example, DNA origami, where we have conflicting requirements to have the canonical base pairing in the scaffold. So how do you see that this challenge could be addressed, for example, in a scalable fashion, not just a simple sensor of just 3 strands, but basically in a longer context where there are longer stretches of DNA sequences? And how do you see that this could be achieved? Do you have any idea?
Oh yeah, well, thanks for the question. Well, I think that when we work with the shared synthetic molecules, you will find that the longer the sequence you have, the more stable will be triplex DNA, and I would not go below even below 12 nucleotides, because then the melting for this 12 nucleotides triplex that I worked with, the melting temperature was around 32 degrees. So which means, well, they will be stable at room temperature, but in a warmer environment like 37 degrees, they will melt. But there is a way to produce shorter triplexes that will be stable. So apparently they like polyamines, and also in the natural environment, like in the bacterial genomes or in biofilms, there are abundant polycationic polyamines. They bind to DNA and facilitate triplex formation even in short regions. I mean, few 5-6 bases. So and this is stable at 37 degrees body temperature. So I would say if you will be building like DNA origami system, and you will like to incorporate a triplex DNA, you may consider to also incorporate some polycationic molecules, such as spermine, or recently we found out that even chitosan that is widely used for DNA mobilization, it's polycationic at pH 6 or below, so it can be used to flip bDNA into different conformations. Well, we used it to flip it into zDNA, but I am like 95% sure that it will also stabilize short triplex DNA.
Okay, great. Thank you very much. Yeah, that's all.
Thanks.
Are there any other questions?
I think everything was clear.
It was a really great presentation. Actually, I had the magnetic particles, so I'm a material scientist, so that makes me wonder, like, for the magnetic particles, like, how if you modify like the particles, like, you know, perhaps like through like a surface modification or something like that, would that cause any like difference to like the biosensing property, or like would that be like, you know?
Yeah, I mean, it's a good question as well. So the size of the particle must be not too big, not too small, so that we can detect the anisotropy. How to say, if the particle is going to be too big, for example, 1 micrometer, then you won't see the change of the clustering by optomagnetic sensor. You may use the other methods like microscopy, for example, and to detect aggregation. But the cool about that one was that it was sort of label-free. I mean, the availability of fluorescence and that's also cost and everything. So we wanted to build something non-fluorescent. And for that, the size 100 nanometer was just perfect. If you immobilize, I mean, modify it, as long as you do not change the size and you preserve the anisotropic shape, like what I call needle shape, because they are not spherical, they're more like ellipsoidal, because the ellipse then rotates, all of them rotate in the magnetic field, and the light is just passing through, and we can detect this light. And once they clustered, they block the light, so we basically detect binding by a blocking light, so to say. So we use the streptavidin coated magnetic nanoparticles. I would assume that other modifications will be also fine. Just use DLS to confirm the size, and you'll be fine.
Cool. Yeah, thank you.
Mm.
Really cool project.
Yep.
If there are no other questions, we can conclude the session. Thank you to Antonio and Emil for preparing these wonderful talks, and we'll have another session in October.
Yeah. Thank you very much, guys.
Thank you. Bye.
Yeah.
Thank you.
See you.